Okay, we are still missing one of your friends, but since I can now record the session, so we'll just make a start, and whoever is misses anything, I will just post again the video link on Spectrum. Okay, so that's normally how I do it. So any recording, I will post it on Spectrum, and then you can just click the link from Spectrum, and then it will open a YouTube, and you can watch the video from YouTube. Okay, so we missed that one. So that's my WhatsApp. Well, hopefully nobody from the internet will text me. Okay, so as I mentioned, for the schedule, we have live session on week 8, week 12, and week 14. And for week 14 is basically is for your presentation. So what I will do what we will do today is we will learn a little bit about HPLC, and then towards the end, I will give you an assignment. So the assignment is related to HPLC so that you can do a little bit of background reading while as we go, and by week 14 you can do your presentation. So ample time to prepare. Um, and the lecture will be more of a active learning. I will try and do it as best as I can. Before this, it will be easier to do it because everybody is in front of me, so we can just have a normal discussion. But since this one is a virtual learning session, so it might be a bit difficult to do it. So I'm planning to do 1 to 2 hours of lecture, and then half an hour of activity, and then from the activity another half an hour for the presentation. But since today the first hour was given to Dr. Mugunda, so we will just we'll just focus on lecture for today, and then at the end I will give out your assignment so that you can start preparing. So there's no activity and no presentation for at least for this week. And to substitute the activity, the discussion and presentation that I normally have in class. Okay, so what I will do is for next week and for all the offline learning session, towards the end, you will need to do a summary and a summary of what you've learned. A very short one, like half a paragraph is more than enough. I will give you the link. I will post the link on Spectrum, and that will be part of your assessment in terms of participation. Okay, so I've written that in class assessment participation. So previously, what I did was as long as you participate in the class, then you will get a full mark for that particular section assessment. Okay, but since we cannot do it now, and of course because an offline session is very difficult, I do not want to just simply have a chat in WhatsApp. So I changed it a little bit. So what you will do is you will write a summary of the lecture. Okay, so it's pretty much what you've learned. Write it down. Okay, so it will also be good as a feedback for me so that I can improve on the content or on the speed of the lecture or on the speed of the way I'm talking, because I understand some of you from international might have a slight difficulty in understanding things that I've said, especially if I said it too fast. Okay, so if that happened, do let me know so I can slow down a little bit. Okay, next one. Okay, so for this course, especially for my section, this is how the marks will be graded. Okay, so the marks will be given. So first off is class participation or class activity, and this will be substituted with the summary of lectures. Okay, except for today. So you don't have to do anything for today. So this will be only for the offline teaching and learning. Okay, but for today, I would say everybody participated. So everybody introduced themselves except for that one person, which I didn't know. I'm not sure if he or she is still registered in the course, but based on the name list that I had, there's actually 14 of you, but now there's only 13 who did registration. Okay, and then moving back to the lecture slide. So the first grading will come from the summary. The second one, there will be a quiz, which consists of 25% mark, and it will be scaled down to 25. So pretty much whatever you get for your quiz divided by 25, that mark will be contributed towards your final mark. Okay, and then we will have a presentation. This one is in week 14. It will be a very short presentation, and that presentation is also related to your assignment. So whatever you had for your written assignment, you will do a presentation as well. Okay, so I do not want to give like two different topics, that towards the end of the day you do need to do a lot of too many research and you don't have enough time. So what I normally do is, I give out a written assignment. Okay, and written assignment normally is due in week 13, and then in week 14, then you'll do your presentation. Okay, so that's my way of helping students to organize, so that it won't be too messy. Okay, so these are the four assessment, or what we call as continuous assessment. Okay, that one, the continuous assessment that we do for the master's course. And normally we'll have 50% of final exam, which we'll divide it into two. One is for Dr. Mugunda section, the other one is for my section. But Dr. Mugunda have already decided to do an alternative summative assessment. I'm not sure if he talked about it already or not, but what it means is that instead of having a final exam, your final exam will be something else. Like for example, during lecture session like this, you will have the exam. Okay, so instead of waiting until the end of the semester, you will have it like very early. And your summative assessment for Dr. Mugunda's section, if I'm not mistaken, will be on week 10, was it? Will be on week 12. Okay, so it will be on week 12. But for my section, you will have your final exam. Okay, and the final exam will be during final exam, which is normally in week, so we will finish at week 13, 14 for study week, and then week 15 is week 16 is the first week of examination. So week 16. Okay, so I'm not sure about the schedule yet, so it will be either in week 16 or week 17. Okay, so meaning that either 2 weeks or 3 weeks after we finish up everything, after your presentation and whatnot. Okay, I think that's it for grading. And these are the list of references that you can use to have a basic reading about HPLC, and an additional reading that is not covered in the lectures. Okay, you should be able to find the book online, or alternatively you can also purchase the book online, or alternatively you can always come if you are in KL or around PJ. You can come to the library and you can request for these books. Okay.
Doctor,
Yes.
if let's say I'm looking for a book more on HPLC, which one that you would recommend in these four that you list here?
Well, actually, they are more or less the same.
Okay.
Of course, principle of instrumental analysis is related to both GC and HPLC.
Mm-hmm.
So to have a good background reading, If say you don't have any background reading at all,
Mm-hmm.
then that book from Douglas is the best, because as you also see, it's already on seventh edition. So this team did a lot of revision on their book.
Mm-hmm.
So they have like a very basic introduction of HPLC, and they how the technology moves. So yeah, I would say go for that book.
Okay. All right. Thanks.
Okay.
Yep.
All right. So that one is no longer needed. We'll skip that one. And the learning the learning outcome for this section is pretty much listed in the PDF file that is already being shared in your Spectrum. Okay, so these three that I mark with the red arrow are the learning outcome for this course. So one is you can illustrate the principle of GC and HPLC, which include all those three things: basic principles and theories behind HPLCs, the modes for HPLCs, the components of HPLCs. Okay, and then you also, based on your learning, you should be able to solve problems related to GC and HPLC, meaning that, say, if we give you a scenario of a situation, okay, you should be able to link that scenario to HPLC, either problems or something to do with GC and HPLC. Okay. Additionally, under the banner of solving problem solving comes QA and QC, so quality assurance and quality control. And these are the two basic, I would say, uh, application of HPLC and GC in both industry and in research. Normally, you will have this. Either you have both QA and QC, or you have only QC. And then industrial application, of course, we will probably go a little bit, a few examples on industrial application. And of course, the industrial application will be more on my experience. Okay, I'm not working in a very big industry, but I did work in a research industry, whereby they did a lot of research that involve HPLC usage. Okay, and finally, the learning outcome is for you to be able to interpret GC and HPLC data. So from the data, you should be able to understand what it means. Okay, what does the different peak means, and whatnot. Okay, so let's start from LC to HPLC. So LC is, for those who do not know, is liquid chromatography. Graphy. Okay, so LC is liquid chromatography, and HPLC is high performance liquid chromatography. Okay, so LC, an example is by using column. So for those who are not chemists, if you are unfamiliar with a column, that is an example of a plastic column. So you might use a different type of column. There are glass column, there are big ones, small ones, variable size, um, and basically the basic principle of LC now will be applied to HPLC, and you can do a lot more stuff using that. And that is what you're going to learn today. Okay, and throughout the lecture, you will hear a lot about HPLC is better than GC. Okay, so it's actually a fun thing to say that in a live lecture, but without you guys in front of me, it means nothing. So we will see. And I'm sure you guys have seen, or at least if you're working with a lot of GC or HPLC, you've seen this type of graph a lot. So this is what a graphical data representation of HPLC looks like for those who have not worked with HPLC. And on the picture on the right is one example of HPLC from Agilent. Okay, this is a brand that produces HPLC machine. There are two or three more, probably a lot more different brands, but the ones that we in the chemistry department uses are mostly from the Agilent brand. All right. So what is LC? What is liquid chromatography? So LC is a separation technique. Okay. So that's the keyword, a separation technique. So when you have something mixed together, you want to separate them. That involve a placement. So without reading the red ones first, okay, just read the white one. Placement of small volume of liquid sample into a tube. Okay, remember the plastic tube that I've shown in the previous slide, packed with porous or solid particles. If you actually look at the very bottom, we'll go through this again. Okay, if you remember the picture, at the very bottom, there are some porous or solid particles. You can actually see it's very, very detailed. Okay, and therefore creating a void volume for separation. So you have a column, okay, a column, that's what we call, or a tube, and then you put your sample in, and your sample contains, in the column itself, contains solid particles, whereby there are a lot of variations of particles that you can use, so that you can create a void volume, or create an interaction between your sample and the solid particles, so that you can separate your compound. Okay, towards the end of the day, this is uh, a long version of a separation technique. Okay, and the red ones are more catered towards HPLC. So for LC, normally you put your sample in. So you place your sample using, say, for example, a pipette. But for HPLC, normally instead of manually using a pipette, you will use an injection. Whether you do it manually or you do it automatically, where the machine will do the injection for you. And instead of a tube, like I've shown before, we have columns, steel pack columns, or polyethylene made columns. I will show some pictures in a few slides. That contains these porous solid particles, or in chromatography terms, we call it stationary phase. Okay, stationary phase. So when you have a column like this, imagine this is the same column as the previous picture. You will have a porous particle somewhere right there. Okay, and then you will put your sample in. And of course, because it says liquid chromatography, so your sample will be in a liquid form. If your sample is in a solid form, you it depends on the size of the solid. If the size of the solid is in a nanoscale, then you can still use liquid chromatography. But if your sample is in micron size, then LC is not the best option. Okay, you can still do it, but you need to make sure that your sample dissolves in the solution first before you can run the LC or HPLC. Okay, so moving back to the picture that I've drawn on the left-hand side. So you have a liquid sample you put into your tube, and then you will put in solvent. Okay, this solvent is what we call as mobile phase. Okay, so the liquid, the solvent that you put in is the mobile phase. So now you have two things that you put in. One is your liquid sample. The second one that you will put in is the solvent. The solvent is the mobile phase. Therefore, the solid porous particle are actually your stationary phase. So this is the basic principle of LC. So you definitely need to have a mobile phase, and you need to have a stationary phase. Okay, without these two phases, you cannot separate your compound. So it's a must to have both in your system. Now, so where individual components of molecule of sample are transported along the tube by a liquid moved by gravity, as I mentioned. Okay, so that's number two. Okay, so this system and an LC system move by gravity, or in some groups, they like to use air pressure. Okay, to actually push the whole system. So what they do is they have like a, a tube connecting to an air compressor, so that you don't have to have a gravity, but you can still make sure that the liquid is flowing, okay, so that you can have your separation. So that's the basic technique of LC. um, as a reminder, if at any time you guys are confused with what I'm saying, you can always type it on the chat area, okay, and I will read it, okay. Because again, this is a lecture, it's not a one-way thing. Normally I'll do that for offline, where I'll just talk around alone for 2 or 3 hours, and then that's it. But for this one, because it's a live face-to-face lecture, so you can always ask me during my lecture. Okay, so this is an example of an LC. On the left hand side, that is a glass column, and you can see inside there, there's this white solid. Okay, so up until somewhere there. Okay, so these are filled with the white solid, and this white solid is the stationary phase. Normally, in the lab, we will use a silica particle. Okay, and then the top one over there is the solvent or the mobile phase. That is the picture on the left is the real image of a LC column. On the right hand side is the image showing you how the separation technique works. Okay, so first off, where you have your column with the silica particle colored in yellow, okay, that one, and then where you have a reservoir at the top where you can actually fill in with your mobile phase. So, but before you fill in with mobile phase, first off, you load your sample, and then you make sure that the sample is actually, if you have not done chromatography before this, then it's a bit difficult for me to show it to you because I don't have a column over here. And to draw it is also a bit difficult, but basically the first step that you do once you prepare your column with your stationary phase solid particle is first to load your sample. So once you load your sample in, you add your solvent, and then as the solvent goes out, or as the solvent elutes out from the column, you will need to keep on adding solvent to make sure that the solid particles are not dry. And while the solvent is going out from the bottom, it will also carry some of the component of the sample that you are actually separating. Okay, as shown in this picture. So you have the three colors, blue, green, and red. And as you go, then the first one that will elute out in this example is the red ones, followed by the green, and finally the blue ones. Okay, so is the flow of a liquid chromatography. So this is just a picture to show you how we actually do it in the lab. Okay, and you can see over there, that's the mobile phase. This picture is a bit better. You have your compound A, B, and C. You can see that initially the separation is very minimum, and then as time passes, and of course you need to keep on adding solvent. As you can see, the solvent in this one is until up there. Okay, so as you keep on adding solvent and let it elutes out down there, you can actually separate the compound, and finally you can get all the compounds separated in individual component, in individual tube. And finally this tube, of course depending on the physical properties of the original sample, if it's liquid, then because now it's a mixture between your mobile phase and your sample, so what you're normally interested in is to evaporate. So you use a rotary evaporator so that you can get a pure sample, pure concentrated sample for whatever purpose that you want to use afterwards. Okay, so the component of the sample are separated by column packing porous solid particles that involve various chemical or physical interaction. Okay, so we will have a lecture specifically on the column materials afterwards. Not today, of course. In a few in a few weeks. Oh, so that's the last person. Let me just pause for a while. I need to get the Google link again. So for the ones that just entered, in the chat area is a link to Google Doc. So can you please sign in as your attendance from using the Google form? Okay. All right. So and also, sorry.
Doctor.
Yes.
This one question. This pack column still using until this now, or is it just some knowledge?
Sorry, can you repeat the question?
Is it the LC column? Is it the LC technology they say use it until now, or just
Yes.
for?
So normally this LC column is being used for a large scale separation. So say for example, if you have 100 grams of compound that you want to separate, then you will normally still use LC because HPLC, even though you can do a larger scale, but you need to have a bigger equipment that can support the big separation. And I'm not sure about the industry in Malaysia, but the research institute that I work at back in Australia, they do have a column that can separate 50 grams of compound. So it's kind of like pre-industrial, so it's not like a very large scale. So we still use it, but that one is HPLC system. But for LC system per se, it's normally on the research level. So when you do research, normally you want to make sure that all the compounds are separable. Okay, so if you know that the compound are separable, then you can move to HPLC for automation, and so that you can easily get your compound. You can still use HPLC straight away from scratch, but in the research perspective, there are still limitations that you need to consider. For example, research grant. So if you are working in a lab that has a lot of grants to buy chemicals, to buy the mobile phases, then yes, you might switch to HPLC from the start. But otherwise, in most chemistry lab, when we do a separation, we will start off with LC, then we might move to HPLC. Okay, I hope that answers your question.
Doctor, right. Because I have experience doing the LC column during my undergrad.
Okay, so we will still for the undergraduate students, we are actually still teaching them on how to use LC, but the LC is not a silica type LC. It's more of ion exchange LC, but the principle is the same. It's just that the particles that is being used is different. Okay. All right. So chemical and physical interaction between molecules and their packing particles. So we will look into this in more detail in the column lecture. Okay. So for now, I'll just skip through about the interactions and whatnot so that it doesn't get too heavy, because the plan is for us to finish by 8:30, and I'm not sure if we can finish by 8:30 if I keep on holding back and speaking about everything. Okay, so and the second point is the separated components are collected or eluted at the end of this column and identified by an external measurement technique such as a spectrophotometer that measures the intensity of a color, or another devices that can measure the amount. Okay, so this second bit is part of the assignment that you guys will do. Okay, but of course, the component will be related to HPLC, not just LC. So LC is, as shown previously, is the column. So you have a column, you elute out your sample, and then you take your sample and then you do analysis. But for HPLC, what you can do is, because the whole system is automated, then you can have the measurement equipment connected to the HPLC. Okay, I will show you some pictures about this soon. Okay, again, this is just a picture of liquid chromatography, so I think we can just skip this. And what is HPLC? HPLC is high performance liquid chromatography, as I mentioned. It can also be referred to as high pressure liquid chromatography. So it depends. It's like a chicken and egg. It doesn't matter. We just call it HPLC because everybody knows HPLC regardless of the name, and everybody knows HPLC is an excellent instrument for compound separation. Okay, so HPLC have been around more than 35 years and is the largest separation technique used, especially in research and in some part of industry. There are different techniques being developed now to actually skip using HPLC, but towards the end of the day, if you do not separate, if you don't use HPLC to separate your raw component or your raw sample into a fine sample, you still need to use HPLC for two purposes. One is QA, quality assurance, and another one is QC, okay, quality control. So regardless of where you are, as long as you are in the industry that produces something, either you produce it, or for some of you guys who are working in the oil industry, oil and gas industry, you will still need to use at least QA and QC in your analysis, and definitely you will use HPLC, either HPLC or GC. But for me, if I were to compare between GC and HPLC, I always go to HPLC because regardless of what your sample is, normally you can analyze it using HPLC. But of course, GC is cheaper, it's faster, and it's less expensive. Okay. So this is the HPLC that normally I bring. The HPLC that I normally bring the student to. Okay, so this is in chemistry department at the university. So we have this brand where normally I will let the students have a go, play around, touch a bit, few things, but this is what we have. So basically the HPLC components contains one, a reservoir. Okay, so this reservoir is for to keep bottles containing the mobile phase. Okay, the reservoir is not the mobile phase itself. I'm not sure. Okay, never mind, we'll try and finish as soon as possible. Okay, and then second one is we have a degasser. For this is just an advertisement, just advertisement for a while. For the Muslim who are going, who need to pray for your Maghrib, you can just go and pray. Okay, in my case, because I'm not at home, I'm actually a Musafir, so I can just do my prayers after the lecture. But for you guys who need to pray because you're at home, so by all means, you can just skip for a while, go and have your pray, and you can always go back and see the recording afterwards. Okay. All right. And then second, as I mentioned, the second component is a degasser. So what it means is that when you have a liquid, there's always a dissolved air component inside the liquid. So a degasser actually removes these dissolved gases so that when it goes into the machine, when it goes into the pump, it won't form any bubbles inside your tubes. Okay, so bubbles, you don't see any bubbling issues in GC, but in HPLC, bubbles are one of the primary issues that people who use HPLC will encounter. Am I correct, Rahman? Have you experienced this before? No, if Rahman went to prayers, then it's fine. Okay, so what we will do next is also to look at each component in a bit more detail. So for now, I'm just going to go through in very brief. Okay, so next one, the third component is a pump or a mixer. So what it does is for HPLC, you can have, if you can see over here, you can at least see 3 bottles, right? So what it means is that for an HPLC machine, you can have more than 2 different solvents at one time. So you can have a mixture of a few solvents. In this machine, this particular machine that I'm showing you now, can actually hold 4 different types of solvents or mobile phases. Okay. So say for example, if you are working with a polar component or an unknown sample, for example, so what you can do is you can load the sample into the machine, and then you can try and have a guess of a mix of solvents. So you can probably choose a 25% of solvent A, and then 25% of solvent B, and then 50% of solvent C, 0% of solvent D. So you can do a mix of mobile phases percent, and you will see how it separates your compound. So that's the beauty of HPLC. You can do a lot of different types or different gradient of solvents very, very easily. As contrast, if you do LC, then it will just focus on normally a set of a set concentration of mobile phase. You don't try and mix it as you go. Okay, but HPLC, the advantage is you can change the solvent concentration as you go. And of course, being that I'm teaching HPLC, if you were to compare with GC, it's kind of like changing the temperature of the whole system, which you can't, because normally you need to know what are the temperature range for your compound, because if the temperature is too low, then your compound will not be volatile enough, then you cannot detect it. But if it's too high, you can also have a chance of burning your compound, that towards the end of the day, you cannot see it as well. So HPLC, the beauty is you can do this compared to GC. So if say your company asks you, they have a set of money and they want to establish a GC or LC system for QA and QC, which one should you choose? So the obvious answer is HPLC. Okay. All right. So next to the mixer, then you have an auto injector. So auto injector is a big storage whereby you can keep your samples. This auto injector can, this one specifically, can keep up to 100 samples. But depending on the type of auto injector that you have, some can keep up to 200 samples or even more. Okay, the bigger the auto injector, then the more samples that you can keep. Additionally, the auto injector, for some of them, they can be refrigerated, meaning that if say your sample is easily degraded, then you can keep it, set the temperature to say about 20 is to, it's like room temperature. So maybe 4 degrees Celsius or 5 degrees Celsius, to make sure that your sample is not degraded as you analyze it using your HPLC. So if you were to draw the flow of solvent, so the solvent comes from the reservoir up there, move it to the degasser, so that it can remove all the soluble gases, and then it goes to a mixture or pump. So depending on number of solvent that you have, you can have a mixture like that, and then the mixture goes in into the auto injector. So you can have an auto injector that injects your sample into the solvent flow, and then instead of going up, it will go down here into the oven. Okay, so you move to the oven. This is where you have your column. So this is where you have your stationary phase, okay, or your column. Okay, and for this column oven specifically, it is also an oven, so you can change the temperature, and by changing the temperature you can get different results or different separation. As a chemist, you know that if you give heat to any molecule, then the molecule will move faster. So same principle applies here. Um, some compounds, because they might be viscous, or they might have a very strong attraction to the stationary phase. You can heat it up so that it moves faster. So once it moves faster, it stays shorter time on the column. Okay, so that's why some people will use column with an oven. So it's kind of like a bit weird, because at one point auto injector you cool down everything, and then on the oven you heat it up. But that's what we do, um, because sometimes you need to cool it down so that you can avoid your sample from degradation. But then when you want to analyze it, you want to analyze it as fast as possible, um, so that you don't have to spend a lot of time to actually separate the compound. Okay, once it goes out from the oven, it will go to the detector. So this detector, I can't remember the code name, but this one detects UV and visible light. Okay, so only compounds with chromophore and a double bond, unsaturated bond, can be detected using this machine per se. But there are a lot more detectors that you will do, that you will learn, and that will be part of your assignment. Okay, so this is the component that you will do for your assignment, for your written and your presentation assignment. Okay, once it goes out from the detector, it won't go into the bus communication, because bus communication is pretty much like a computer. So it has no liquid in it. So it will go out from the detector. That one is a fluorescence detector, but we hardly use it anymore, because the compounds are hardly fluorescence, and the project that we are doing is not really a fluorescence, more on UV-vis compounds. And then it will go out into the best machine or best component, which is the autocollector. So again, the principle of LC is to separate compound, and then sometimes you want to separate compound and then you want to collect them as well. So HPLC allows you to do that automatically. So you can just set everything in the computer, and then the computer will tell the machine to collect this, and this. So it's very easy. Even you have an auto injector, so you can imagine if you have work in the lab whereby you do everything manually, where you prepare your own column, you do a separation, at the same time you manually collect all your samples. By using HPLC, you can automate everything, and then the free time that you get, you manage to save, you can actually use it for different purposes. Okay, so this is HPLC component. And I hope that you do learn a little bit about the components. And finally, on the right hand side, of course, we have a display or software that controls the whole hardware. Okay, so some differences between LC system, liquid chromatography system, and HPLC system. In principle, LC and HPLC works the same way, except for one, speed, efficiency, sensitivity, and ease of operation. Okay, again, but towards the end of the day, provided that if you are trained in using it. If you are not trained in using HPLC, regardless of how many HPLCs you're given, you will not be able to complete your work. Okay, but if you were trained using HPLC, then pretty much all your work will be blazing fast. Okay, a normal column like this normally takes you from the preparation stage up until you manage to separate everything. And depending on how many compound you want to separate, for example, it's shown here is just two compounds. So normally for separation of two compounds, including preparation of column, this and that will take you at least 2 hours to prepare. Okay, but the same system using HPLC, just to analyze or just to separate the compound, well, the minimum that you can do, the minimum that normally I would do is just 15 minutes. Okay, so imagine you can shorten your time by a lot. So this one is by a factor of 4. Mm, is it 4? 15, about 25%. Okay, 1/8. Yeah, so by a factor of 4. Okay, and imagine that using HPLC as compared to a column. So this one is one sample, takes you 2 hours. So in 2 hours, you can actually analyze 8 samples. So you can imagine if you are working before this using HPLC or LC system, switching to HPLC will save you a lot of time. And of course, a clear advantage is the AutoCollector. Okay, instead of collecting it manually, so if you have an AutoCollector system, you can just ask the machine to auto-collect it, and then you can just run your system overnight. The next morning when you come into the your lab or your office for work, then the compound have already been separated for you. You can just straight away do your analysis. And this system, if you are doing like a PhD, for example, that uses a lot of separation technique, then uh, this is definitely a clear advantage. Because when I was doing my PhD, I learned, I self-learned about using an automatic system like this, and you can pretty much work day and night. So during the day, you can do a lot of synthesis. At night, you can ask the machine to purify it for you. So you pretty much like doing um, 24 hours a day. So yeah, again, if you're working in a company, and if that company does a lot of QA and QC systems or QA and QC analysis, and the company has money to spend on either GC or LC system, then definitely go for that, because it will definitely save you a lot of time and increase your productivity. Okay, so this is just another picture about how HPLC work. I've already briefly mentioned about it in the last previous 2 slides. So again, you have solvent reservoir. The solvent moves to a degasser, and then from the degasser, it goes into your pump. And depending on the system, so you can have your sample injector here, and over here, instead of having a sample injector, you have a mixture. Okay, where of course the pump is also a mixture, so it mixes your different solvent. If you are to run an isocratic flow system, so from one side where you have your mobile phase, on the second side, the other side where you have your sample being introduced into the system, it get mixed up in the middle before it will be sent to the column, being separated according to either the affinity or whatever parameters that you set, goes into the detector where you will see the separation peaks, and then either it goes to a waste, or you can collect it again using an AutoCollector. Okay, and you can reuse the sample. And towards the end, from the collector, the data will be transferred to your display, and you can analyze the area under the curve that normally represents concentration. And of course, because you are using columns and everything is automated, you can calculate the retention time or elution time of your compound. Okay, and this picture is just showing you the solid particles that are being put inside an HPLC column. So normally in the first lecture, I'll bring my own column that I have in the lab and pass it around for those who have not actually used HPLC, so you can actually see the column and you can see that they are different sizes like this compared to that. Okay, and they are different in length. For example, that one is compared to this. So there's a lot of variation, and this one is just the physical variation. There are actually they can be differences in the individual solid particles as well. Okay, there are a lot of technologies out there, and we will learn and touch some of them, not all, definitely, because otherwise, it will be a year-long course. Now, HPLC is a separation technique that involves injection of a small volume. I think this is just more a repetition of what I said. Okay, so it injects a small volume compared to an LC, where LC you can have a bigger volume of sample. Normally you put it in a very small tube. Okay, we call it HPLC tube. The size is about that much, or if you were to show this pencil, it's about that. That is the size of the HPLC tube. Okay, tube with tiny particles 3 to 5 microns called stationary phase. So this one is the column. So you inject your sample again from the auto injector. You inject it into the solvent flow. It goes into your column that contains tiny particles. Nowadays, you can actually get smaller than 3 microns. They are 1.8 microns particle as well. Micrometer, okay, called stationary phase, where individual components of sample are moved along the packed column with liquid from the mobile phase, forcing through the column at high pressure delivered by a pump. Okay, and again, this is the reason why you need to have a degasser in your HPLC system. High pressure by a pump. So imagine that you have a solvent that contains a lot of dissolved gas. So by the principle PV equals to nRT, wait, is this the correct equation? PV nRT. What is the correct equation? I can't remember on top of my head what is the equation that you can use. But as you increase the pressure, you actually promote the formation of gas. Okay, so the dissolved gas now gets together and then form a bigger bubble. So before this, you might not see any bubble, but then under high pressure, it will form bubbles. And these bubbles, if you imagine at high pressure, so for HPLC, you want it to be high pressure. At the same time, the pressure should be consistent. Okay, now imagine if you have a bubble. Okay, imagine if you have a tube. So imagine this is a tube, HPLC tube, where you have a solvent flow. And this tube, of course, is very small, probably around 2 millimeter. Width, the outer diameter, the internal diameter varies. But just imagine if it's say 1 millimeter of internal diameter, where you have a flow of liquid. Okay, and this system is under high pressure. So when you have a bubble somewhere in the middle, what happens when you have high pressure? So the bubble will contract and expand. Okay, so if it contracts and expand, what will happen to your pressure is that, say for example, if this one is time and pressure, okay, what you want is normally a constant pressure like that throughout your run. Okay, because if the pressure goes haywire up and down, your compound separation will also be different each time when you run your sample. Okay, so the pressure can be different and you can still separate your compound, which is fine. But if you were to replicate the run again while having a variable pressure, then the elution time will also be different. So you have a variable somewhere, then your result will also be vary between runs. So that's why for HPLC to work the best, you need to have a constant pressure. So if you have a bubble inside a tube, because it contracts and expands, you can get a pressure like that. Okay, so when the pump pushes, where you have the highest pressure, then you can see the pressure is going up and down. Okay, this is one of the bad things that you will learn in the next few weeks. We will go through in more detail next time. For now, we'll just uh, continue because we have another about 25 minutes. Okay, so these components are separated from one another by column packing. As I mentioned, this is just a replicate. Involve various chemicals or physical interaction between their molecules and the packing material, and we will learn about this under the column lecture soon. Okay, if I'm not mistaken, it's next week, or probably the week after. These separated components are detected at the end of the column. I'm not sure why the picture is coming out. It should be hidden. So what it says here is just that the separated component will exit from the column into a detector, whereby it will be detected one by one. Okay, so that's a simple basic idea for that particular section. I'm hoping that your lecture notes is better than my this probably happens because I'm using an iPad while preparing the lecture notes and whatnot. I'm using a desktop or a PC. Okay, so this is an example of HPLC runs. So 0 minute is the point whereby the sample is introduced into the HPLC system. And that one is whereby you normally see that either from a solvent mix from your sample or an air bubble. When I talk to one of the engineer, they normally say that that one is just a signal sign. But what kind of signal, I have no idea because they didn't tell me. But normally I noticed that this particular signal at the very beginning changes as you change the volume of sample that you introduce. So the more sample that you introduce, normally the signal varies. And of course, it also depends on the type of solvent that you have mixed together with your sample. So there's a lot of variation. It's just that everything will come out like very early, and you will definitely see it if you are doing HPLC. Now, what you will see also is a straight line like this, okay? But in reality, if you actually zoom in to this line, what you will see is something like this, okay? It's a signal signal or noise. And having a small signal to large signal to noise ratio will get you a better reading. Okay, so HPLC is actually not a smooth line as what you see now. It's actually a variation or a signal thing that you will learn soon under the detector section. Now, if you have this compound on the right hand side mixed originally in one HPLC sample tube, and then when you inject it, what will happen is because these compounds have different functional groups, therefore they have different interaction with the stationary phase, and thus they will be separated differently at different times, and this is what you will observe. Okay, so these are called chromatographic peaks. So that's just the terminology. The peaks, or you can just simply call it peaks. Okay, and each one represents a separated component. Of course, similar to GC, you do have variation in terms of whether the peak are sharp, whether the peak are broad, whether the peak are skewed towards one side and whatnot, which um, has almost similar principle to GC. Okay, where it's just that this one is HPLC, that one is GC. Signal, the idea and everything are mostly similar. Thus, there are some parts that I will not touch about chromatogram, liquid chromatogram, because it's very similar to chromatography, gas chromatography chromatogram. Okay, so what can HPLC be used for? Different from GC, HPLC is optimum for separation of chemical and biological compounds that are non-volatile. Of course, you can also use a volatile compound, provided that you can actually dissolve the volatile compound in a solvent, whether it's organic solvent or aqueous solvent. You can definitely run HPLC. Okay, typical non-volatile compounds are, this is just an example, salts, protein, natural products, whatever it may be. You can extract a product from, say, a tree, and then you can actually analyze the component of the products or natural products from the tree. Okay. Thermally unstable compounds such as TNT, enzymes, and whatnot can also be analyzed using HPLC, because, again, towards the end of the day, for HPLC, you can have a chiller. So in the auto injection section, you can have either a chiller or a fridge, okay, where you can cool down the temperature so that it's not um, hazardous anymore. Like TNT, it can be TNT is explosive, so in the presence of heat, a lot of heat, you can explode. So you reduce the temperature so that there's risk of explosion is less. And of course, enzymes, they degrade very, very quick. So having a chiller or a fridge avoids the enzyme from quickly degraded. Even though we have an oven, but the use of an oven is not compulsory. So you can always run your HPLC without an oven, so there is no issue at all. So that's why, towards the end of the day, HPLC is better than GC. Okay, so HPLC is widely considered to be technique mainly for biotechnological, biomedical, and biochemical research, as well as for pharmaceutical industry. Comprises only about 50% of HPLC users. I'm not sure where the book gets this information from. I'm just putting it there because I don't have my own statistics. So the best statistics that I can get is from the book. So that is what the book says. If you do have a better figure, then by all means, correct it. But this is not part of the important things that you need to know about HPLC. This is just like a advertisement. Currently, HPLC is used by a variety of fields, including cosmetics, energy, food, environmental industry, oil and gas. Okay, of course, cosmetic falls under pharmaceuticals. It can also be part of product development. So there's a lot of stuff. It's not just limited to whatever I've written there. Okay. So this picture is showing you the difference between GC and HPLC. So if you have a compound, what would be the best thing to use? So this one is HPLC. That one is, you have GC. Now, for those of you who have used HPLC before, you do know that hydrocarbons like C12, C6 to C2 to C6 hydrocarbons can also be used for HPLC. They are from the volatile gas phases. So actually, the graph can be redrawn looking like this. Okay, so pretty much anywhere around the world, you can just scrap away your GC and then just use HPLC. There is no issue in doing that. It's just that GC is faster. in some places, not every places, but in terms of the samples that you can use, so far I have yet to find a sample that cannot be run by HPLC. The only ones that cannot is the one that are big in size. Like for example, if you have a carbon nanotube, so it is very difficult to actually run it on HPLC because the nanotube can be very big and it can definitely clog your either your filter or your column. Okay, but otherwise, if you have small molecules, whether it's volatile or non-volatile, as long as it can dissolve in the liquid, of course, the liquid cannot be a volatile liquid as well. Okay, as long as you can dissolve it in any usable liquid for HPLC, then you can definitely use it. So these are no longer part of the issue. So regardless of polarity, you can run HPLC. Regardless of volatility, you can run HPLC. So if someone asks you which one is the best, HPLC or GC, your answer now should be HPLC. Okay, so remember HPLC is always the best. Okay, just a tiny bit about quantitative analysis. It's the most common parameters for compound identification in is the retention time. I'm sure everybody knows because you have gone through GC lecture. So this image should be part of what you should now be able to read. Okay, so that is just showing you what happened as either when you have a different products, okay, a different mixture. For example, if say initially you have a leaf and then you pre-process it into a tiny particles and then you dissolve it in a liquid, okay, and then from this liquid, because you can still see a lot of particles, so you might use a size exclusion chromatography or a solid phase chromatography, SPE. Okay, these are some of the chromatography that chromatography or column that normally people use in research or in product analysis. Okay, and then from the SPE you might get a different concentration of samples. So you might get at 10% solvent you get product A, and then at 20% you might get product B, and then at 30% of whatever solvent, this is just a theoretical, you get product C. And then you want to analyze what are the difference between product A, B, and C. So you can run it on HPLC. You can use the same program. The only things that you need to change is you need to use different cubes. So this A will have its own HPLC tube, B will have its own one, and also C. And then you put all these three tubes into HPLC. They will run it for you, and then you will get a graph something like this. Okay, and then from here you can actually see that for example, that one is product E, contains a higher amount of camphorol and isohamretin. Okay, I can't really see the writing. Okay, but you can actually see that product E contains the highest amount of the control compared to say product A. Okay, so this is part of what HPLC can do for you. All right, so this is just another example. Next one, it talks about peak height or peak area. So peak height or peak area corresponds to the quantity of a sample. Okay, so oops, the quantity of a sample. So if you do have a bigger peak, but towards the end of the day, it's not just a peak. Okay, it's area under the curve. So peak heights plays an important role, but towards the end of the day, the area under the curve that matters. Okay, area under the curve talks about the quantity of the sample. So the bigger the area under the curve, the more samples that you have. But peak heights talks more about purity of the sample. So if you have a very narrow compound but with a very high peak, so it just means that the compound absorb more light. But depending on the shape of the peak, you can say that whether the peak itself is from a pure sample or from a mixture of sample. Okay. And this is just to highlight which section is this. I can remember. Yeah. Anyway, we can just skip this. With concentration of X. Oh, okay. So this one is representing the component of I can't remember. Never mind. I'll just skip this for now. It's not really important. It's just showing you what HPLC can do if you have different products. Okay, so preparation of a pure compound, purification can also be done on HPLC. So the figure, the slide on the previous page is showing you about how HPLC can be used for quality controls, okay, for quality controls or for analysis of sample. But at the same time, what you can do for HPLC is doing a purification. So what does it means? So if say, for example, you manage to separate a compound, something like that, okay? So what you can do is you can set the system so that it auto collects the sample at particular time frame. So if that one is, for example, 1 minute and 2 minutes, you can set the system so that it collects between 1 to 2 minutes, and then stop collecting between 2 and 3 minutes, and then start collecting again at 3 to 4 minutes, and so on and so forth. So by doing this, what you can get is you can get a pure sample that towards the end of the day, you can either evaporate to concentrate the sample or freeze dry it and whatnot. And then you can do whatever you want after that. Okay, it doesn't mean that you need to use it for animal testing. You can always use cell for toxicity testing and whatnot. So that's it for this slide. So purification. So first one is analysis. Second one is for purification. So this slide is just showing you some example of how HPLC is being used in the industry. So for chemical, it's mostly on chemical analysis. Pharmaceuticals, analysis, purification, and whatnot. These are just some of the examples in that particular industry. The third one is bioscience, where you can do a lot of protein purification, protein identification. Same principle goes for peptides, nucleotides, sugars. Okay, but sugars falls under consumer products, but same thing. Okay, lipids, antioxidants, clinical amino acid, vitamins, homocysteine, and COVID vaccine. Some of them, the ones from University of Queensland, they are producing a protein based vaccine, which definitely will use HPLC as part of the purification process. Of course, it's not the industry. During research, they use HPLC. But for industrial purposes, they might use something different approach. And of course, finally, for environmental, you can always use HPLC. So for those who are not a Malaysian, so you might not know the issue that is going on multiple times towards the past few months. So our water in Malaysia or in Selangor, one of the states in Malaysia, keeps on getting polluted every month. Okay, so how do they know? Of course, they have a sensor. But as a researcher, how can you identify what are the pollutants? So you can always use HPLC to actually quantify what are the pollutants. Okay, and this is towards the last slide. So this is just an example on how the separation works. Okay, so you have basic system, detector, your column in the middle, your injector, mixture, and pumps are all written there. So you have your stationary phase, which is the column pack in the middle, and the mobile phase is over there. Okay, so this is what happened. So you can actually see as the compound separates, the chromatogram is going bigger and bigger. And once the compound reaches the chromatogram, you can actually see the peaks is now observable. Okay, and this is how HPLC separation works. And this is how you will see if you actually run HPLC. Okay, I'll just show you. Well, it's just a slide. Okay, will it work straight away? Yeah. So you can see again, as the compound reaches the detector, you can see the chromatogram picking up the signal and showing you the area under the curve. Right, GC separation technique. Say for example, I have uh, two separation techniques in my lab, HPLC and GC. Which one should you use? Of course, now you should know what are the answers. Can you guys, whoever is still listening, write down what would be your answer if you have this question? Which one should you use, a GC or HPLC? Anyone still here? Or am I talking alone? HPLC, thank you, Carmen. Yes. Okay, so you always use HPLC. Again, a comparison, same slide. I think we will stop here, and we will talk about a bit more on comparison of GC and HPLC next week in offline mode, of course, because we are short on time. Okay. Thank you, everyone. I will post this video link, the video recording on Spectrum, and I will see you guys again face to face like this in week 12. Okay. Thank you. See you guys. Thank you, everyone. Have a good weekend. Bye.